Now we can see the Sanger's method of sequencing. Okay, we can see the Sanger method of sequencing. Okay, this Sanger's sequencing is also known as enzymatic method of sequencing. Okay, this Sanger's sequencing is also known as the enzymatic method of sequencing, and this method was developed by Frederick Sanger and his colleagues in 1977. Okay, this method was developed by Frederick Sanger and his colleagues in 1977. And this method of sequencing, that means Sanger's method of sequencing, is commonly referred as chain termination method or dideoxynucleotide method. Okay, this Sanger's method of sequencing is commonly referred as chain termination method or dideoxynucleotide method. Okay, we know that dideoxynucleotide are the modified nucleotides that lack hydroxyl group and on the both 2' and 3' carbon of the sugar of nucleotide. Okay, that DDNPs means dideoxynucleotide are the modified nucleotides that lack hydroxyl group on both 2' and 3' carbon of the sugar. Okay, 2' and hydroxyl group in the case of DDNP. Okay, here you can see this is the DNTP. Okay, DNP, that means deoxynucleotide. Okay, this deoxynucleotide is present in the normal DNA. Okay, deoxynucleotide is present in the normal DNA. So you can see here in the second position is hydroxyl group is removed in the case of deoxynucleotide, and but in the third carbon, the third carbon, there's a hydroxyl group is there in the deoxynucleotide, but in the second position that hydroxyl group is removed because or is removed in this deoxynucleotide. Okay, and we told that dideoxynucleotide is a modified nucleotide that lack hydroxyl group on both second carbon and third carbon. Okay, second carbon and third carbon. You see here, there's a second position and there's a third position. What is the problem with that? If there is no hydroxyl group in the third position means, okay, there is no hydroxyl group in the third position means when we are adding nucleotides into the in the PCR reaction mixture, that time when this dideoxy is attached into the chain of DNA. Okay, when this dideoxynucleotide is attached into the growing chain of DNA, that time this 3' OH is not present in this dideoxynucleotide. Okay, dideoxynucleotide. That's why the next nucleotide cannot able to join with this preceding nucleotide because the next nucleotide will be added into this nucleotide by the OH group of this 3' OH group of this nucleotide, because in this dideoxynucleotide there is no OH group in the third position, so it cannot able to make phosphodiester bond with the coming nucleotide. Okay, coming nucleotide cannot able to make phosphodiester bond. That's why that will terminate the chain. Okay, that will terminate the chain. Here you can see these are, for example, this is our template DNA. Okay, this is our template DNA, and this is the primer. Okay, this is the primer. Okay, this is the primer, and we'll add this when we add this normal deoxynucleotide and a DDNP mixer. That is when after this into this primer, when this dideoxynucleotide, this abnormal nucleotide dideoxynucleotide is attached into this primer. That time is this dideoxy does not have any 3' OH group, so the coming nucleotide cannot able to join with this. So the chain is terminated. So this chain is terminated. Okay, this is a chain is terminated. That's why it's called as a chain termination PCR. Okay, chain termination PCR. Here you can see this is another one case. Here this is the up to T is the primer, and in that then in the system that added normal guanine nucleotide, normal guanine nucleotide. So it having a 3' OH, 3' OH, but the polymerase and in the next the adding of the next nucleotide that added abnormal adenine nucleotide, abnormal adenine nucleotide. So here there is no OH group. So the no OH group. So the polymerase cannot able to join this next nucleotide into that. So this chain also terminated. So this type of PCR is called as a chain termination PCR. Okay, chain termination PCR. Okay, this is called as a chain termination PCR because this chain will be stopped. Okay, this chain will be stopped. That's why this sequencing, because this sequencing need this chain termination PCR. That's why this sequencing also, that's why it's also called as a chain termination method of sequencing. A chain termination method of sequencing. Now we can see what are the steps involved in chain termination sequencing or Sanger sequencing. Okay. Now we can see what are the methods, steps involved in the Sanger's method of sequencing. Okay. Normally, like in the Maxent-Gilbert method, this sequencing reaction also take place in four tubes, and this four tubes contain template DNA, DNTPs, that means normal nucleotides, and contain polymerase, DNA polymerase, and labeled primer, okay, radio labeled primer, and any one type of DNTPs. Okay, any one type of DNTPs. Any one type of DNTPs means in one tube is may contain one DD ATP, in other tubes contain DD TTP, and in another tube contain DD CTP, and another tube contain DDGTP. Okay, and the DNA polymerase start to add DNTP to the chain until it's happened to add a dideoxynucleotide instead of a normal one. Okay, uh, when we kept this reaction tubes, okay, when we kept the reaction tube or reaction, that time this polymerase will add DNTP into the chain. Okay, this polymerase will add continuously add DNTPs into the chain until it's add a dideoxynucleotide instead of a normal one. Okay, until it's add a dideoxynucleotide instead of a normal one. At that point, normal, okay, at that point no further nucleotide can be added, so that strand will be end with dideoxynucleotide. For example, you can see here these are template DNA. Okay, these are template DNA, and these are primer. Okay, this primer will be marked labeled, and these are template DNA. And here you can see primer that attach, first it's attached to one nucleotide, nucleotide, that's what the polymerase added nucleotide, that is a dideoxynucleotide, that is a dideoxynucleotide. So that growth of the chain is terminated, the growth of that chain is terminated. And here you can see that in this chain you can see added first one guanine nucleotide was, yeah, this likely this guanine was a normal one. Okay, this gone was a normal one, so this allow to add the next nucleotide. Okay, allow to add next nucleotide, but this is a nucleotide, but this a is a dideoxy, but that's why is cannot able to add next nucleotide to this one, so this chain also terminated. Here you can see add a normal sequence here, and is the second one also a is a normal, but c was abnormal. There's a dideoxynucleotide, so the growth of the chain also terminated. Here also you can see, here you see this is a primer, and here also added one guanine normal and adenine normal, and C is a normal, T is normal, G is normal, A is normal, and A is normal, G is normal, C is normal, but T is a dideoxy, so the termination is okay here. So when we are doing this reaction, okay, for tubes we'll do the reaction, we'll get a, fragments with a varying length. Okay, we'll get a fragment with a varying length, we'll get a fragment with a varying length, and that DNA, that different length DNA separated by polymerase polyacrylamide gel electrophoresis, and the band will be identified by based on autoradiography. Okay, that means we'll separate this different fragment of DNA using polyacrylamide gel electrophoresis, and will expose this gel for a autoradiography, because that primer is labeled with the radial labeled, so we'll expose this gel for autoradiography, and based on the bands in autoradiography we can construct the sequence. Okay, based on the bands in the autoradiography, that we can construct the sequence. Okay, that means we can identify the sequence based on the bands in the autoradiography. We can see here this figure explain how we'll do this Sanger's method of sequencing. Okay, we can see how it will do the Sanger's method of sequencing. Okay, here you can see we are taken template DNA primer plus polymerase. Okay, we are taken uh, template DNA primer polymerase DNA polymerase and this normal DATP, normal CTP, DGTP and DTP, and make a mixer of reaction. Okay, make a mixer of reaction. Okay, make a mixture of reaction with a template DNA primer will be labeled. You have to remember that primer should be labeled, and the DNA polymerase, deoxyadenosine triphosphate, DCTP, DGTP and DTTP. Okay, and this is a reaction mixture. From that we have to divide this mixture into 4 tubes. Okay, we have to divide this mixture into 4 tubes, and in that tube, the first tube will add DDATP. Okay, DDATP will add DDATP. Okay, DDATP means dideoxyadenosine triphosphate, the first tube, and the second tube will add dideoxy CTP. Okay, dideoxy CTP, and we'll add DDGTP. Okay, DDGPTP, and the fourth tube will add DDTTP. Okay, but we'll add DDTTP. Okay, you have to remember that when you are adding DDNTPs, that means dideoxynucleotide phosphate, that concentration should be less compared to our normal DTPs. Okay, that means the concentration of that DDNTP should be less compared to our normal DNTPs. Okay, normal to normal DNTPs. Okay, that means this DDNP concentration should be less compared to our normal DNTPs. And after that, we'll kept this 4 tubes for reaction. Okay, we'll kept this 4 tubes for reaction. In that case, go for amplification. Okay, go for amplification, and that amplification vary in length. Okay, that amplification will create DNA with varying length, because in the previous slide we are told that when at the time of amplification, the polymerase will add nucleotides. Okay, deoxynucleotides into the chain. Okay, deoxynucleotide into the chain. When a dideoxynucleotide is added into the chain, that time that chain growth will be stopped. Okay, that polymerase will add nucleotides into the primer, here for example, here you see this is the template DNA and this is the primer. This is the primer. Okay, and here you see that this ATP, then CTP and GTP and TTP, and this also separate, this reaction will be occur, the same template DNA will be amplified. Here the amplification will be terminated by DDATP, and this tube the termination will be this amplification will be terminated by DDCTP. Here that amplification will be terminated by DDGTP, and this tube this amplification will be terminated by DDTTP. Okay, when adding nucleotides, the polymerase will add nucleotides into this primer. Okay, into this primer. That time, sometime randomly, this polymerase will add this DDNTPs, that means dideoxynucleotides into this primer. Okay, that time that termination will be occur. Here you see this is the primer. This is labeled the primer. Here in G tube. First that polymer is added DDGTP. Okay, DDGTP. So that growth of this chain is stopped. So okay, that growth of this chain is terminated. Okay, but in the case of A tube, that's why here you see that growth occur and first added when one normal GTP. Okay, normal guanine is added into guanosine triphosphate is added, and after that in that A tube polymerase added one adenine, but that adenine was dideoxyadenine. So that chain is stopped here, and like that, this all the case also is there. Here also you can see one C is the C here. See that first in the C tube. Okay, C tube first is added guanine and adenine as a C normal one, D normal one, G also normal one, and A also normal one, but and G this G also normal. But here C random the C was dideoxy. So that termination is occurred, and this adding of this dideoxy is the random. So that will get DNA fragment with varying length in different tubes. Okay, we will get DNA fragment with varying length. In this, I told that in this tube, this DD-ATP will terminate the chain. Here, the DD-CTP will terminate the chain, and here DD-GTP will terminate the chain, and DD-TTP will terminate the chain. And after the reaction, okay, we will get varying length of this DNA in this 4 tubes because of the chain termination of the method of DD chain termination due to the DD-ATP. That's why we'll get varying length of DNA, and we'll find out the length of DNA in the piece in this reaction mixture. For that, we'll load this 4 samples in separate wells in a polyacrylamide gel electrophoresis. Okay, and we'll do this for polyacrylamide gel electrophoresis, and we'll find out which well having the first band. Okay, which will have the first band. Here you see from here to we get a small fragment will be here and larger fragment will be here, because in the electrophoresis this side will get a small fragment. Okay, that's why, because the small fragment will move faster. Okay, and here you see in G tube is after the running the this gel, and we'll expose this gel for autoradiography, and in that autoradiography film we will find the bands here, and in this G tube we found one bands here. So that tube, that sequence G, and because in this tube this band, this strand, small strand, that fragment is terminated by G, the DD and DPG. Okay, so G is there. Here you can see the same corresponding strand. You can see here this is the primer, and here you can see G is attached to the primer. So that band, this chain is stopped. And next of this one we have see here this A in the A tube. You can see so here this sequence is A. So you see here this when is A come here, you can first is add the normal G and is added A. Okay, so A was dideoxy, so it's terminated. In this case you see here this is C tube. Okay, and the C tube is came band is in the C tube. So this DNA is this strand and nucleotide is a C. Okay, C is the, so here you see that first a G normal G is added, but in the A this, and second normal A is added, and but C was a dideoxy. Okay, dideoxy. So that here sequence was C. So that's why C is added to here, and here you see the T. Well, you see in the band. So here see the T is terminated this chain, and here see G in the and this well of G you see here, and here this fragment is terminated by G DDGP. Here is terminated by A, and here is terminated by G, and here is terminated G, and here is terminated by C, and here what is it? Terminated by D. So from this length of different fragment length, we can identify What are the sequence that's present in our DNA? What are the present in the DNA? So here you can see G A C T G A G C T. This is the sequence. Okay, this is the sequence, or this is our sequence. So this is the complementary strand. Okay, this is a complementary strand. From this we have to find out what is the original sequence of our template, because this is the sequence of template. For that we have to convert into this is a complementary string. Okay, this is our original template string. So this is a template string. So when we'll get we'll run this gen, and autobiography will identify the sequence present here, G A C T G A G C T. So its complementary will be the our template. This DNA's complement DNA sequence complement will be all the our given DNA template, or the given DNA template. Yeah. G A G A C T. This is the Sanger's method of sequencing. Okay, this is the Sanger's method of sequencing. I hope you understood this. Okay. So I hope you understood this DNA sequencing. Thank you. Thank you for listening.